I'll get a rubric put together, and I'll figure out the podcasts that you are to listen to and evaluate using this rubric. What else? Friday. Oh, did anybody has anybody looked at their results for the Cocoa Lab yet? Amazingly, the data worked for the class. It worked. I shouldn't be shocked, but I was kind of surprised that it worked as well as it did. So good job. I haven't fully processed the data myself. That's your job. But just looking at the raw data, it's pretty interesting, I think. So you can dig into that data this week. And the questions are on the Moodle. They've been there since last week. And you can answer them and process your information. You'll need the data that's in the class spreadsheet that's there in the Moodle. Friday, we're doing, hopefully, a maple syrup analysis lab using 40 different samples of maple syrup from Travis's farm. And then I'm going to bring a couple maple syrup unknowns. Unfortunately, Travis's maple syrup comes in these incredibly teeny tiny little vials, so we won't get to taste any of that, really. Maybe the tiniest taste, we'll see. Yes? I have some. Sure, yeah, bring some of his maple syrup in. Yeah, if anybody's got a weird maple syrup they want to analyze, needs to have enough so that the whole class can get a taste, so we can have tasting notes for the class on that maple syrup, and then we'll run the sample and see how it passes through our little chemical filter process. So that's Friday. And then the last Friday, the 8th or whatever, we will basically bake cookies go through the same process that I was describing for pie crusts with cookies. We're looking for the perfect sugar cookie recipe. We're going to try all different ways of making cookies. There's probably going to be some gross ones, maybe some good ones, who knows. Hopefully there will be some excellent cookies. And we'll also clean up the lab. That'll be exciting. All right. Anything else do I want to say? I'm skipping, well, I'll tell you what I'm skipping when I skip it here. All right, so I didn't quite get to give the review of this last unit that I covered, and I'm going to do that now. So this last section was about cells and metabolism. Started by talking about the anatomy of a cell and listed all the different parts of it. The important parts that I talked about were the apical surface, where there were the villi on the surface that increased the surface area of epithelial cells, and then the nucleus and mitochondria were important because they played into our discussion of metabolism later. The others were just parts of the cell that are important but not especially germane to our discussions. I talked about the 4 tissue types: epithelial, connective, neural, and muscle. And then I focused on epithelial tissue because that's what lines our GI tract, and that was a prelude to talking about the GI system. I talked about the basic anatomy, starting with the oral cavity, and I talked about taste. And when talking about taste, I described the labeled line code, where each of the different sensory modalities, sweet, sour, salty, etc., was interpreted by the brain because it was traveling along a line that the brain interpreted as salty or interpreted as sweet, etc. The parotid. Then I talked about salivation and the 3 salivary glands: the parotid, submandibular, and sublingual glands. The 2 types of secretions in saliva, serous and mucus secretions, and how they distributed across those 3 glands. And then I talked about the 2 taste loops in the mouth. The presulcal, there's that line, the sulcus, that divides the front and back of the tongues, and there's the presulcal and the postsulcal. Afferent meaning sensory, and efferent meaning motor or salivary loops of the facial and glossopharyngeal nerves. There was a slide in there that talked about that detail. Then I moved on to the stomach. I talked about the histology, or the cellular makeup of the stomach, with the gastric pits that had the 3 cells, the parietal chief and G cells. Parietal cells making HCl, and I talked about the alkaline tide, how carbonate is exported into the blood as we export HCl in the opposite direction into the stomach. We talked about how that regulates pH in the body a little bit. Talked about the chief cells, which produce the zymogen pepsinogen, which gets turned into the active protease pepsin by HCl, and then the G cells, which produce gastrin. That led us into talking about the way the stomach works, the three phases, the cephalic, gastric, and intestinal phases. That's sort of the look and smell, the actual eating, and then the moving on into the intestines phase. I told the story about Barry Marshall and the Helicobacter pylori in gastric ulcers. Then into the small intestines, I talked about the morphology, the high surface area with the plicus auricularis and the villi, which give us the high surface area for absorption, the three parts of the small intestine. I talked about the pancreas, the endocrine and exocrine cells, the endocrine making insulin and glucagon, the exocrine making the four types of enzymes that help us break down all the nutrients that we need. I didn't really talk about deoxyribonucleases or the lipase much, but I talked about amylase and how saccharides get broken down, sort of reprising what we had talked in the previous unit. And then I talked about the proteases and peptidases, defined what a zymogen was. Focused mostly on trypsinogen and its transformation to trypsin, but there was others in there, carboxypeptidase and chymotrypsinogen. I didn't say much about the liver or the colon, but then I moved into the absorptive and post-absorptive states, which was a way for me to talk about metabolism and how we're actually using energy, the nutrient pool, anabolism, building things up, catabolism, breaking them down, the TCA cycle or citric acid cycle, and the electron transport chain as the two mechanisms in the mitochondria for making ATP. Then I kind of ran out of time before I got to talk about ketone bodies, and I'm pretty much skipping the talk about the hormonal control of appetite, leptin, and ghrelin, and what metabolic syndrome is. That's okay. We don't need to have it. Are there any questions about that? That's sort of a detailed outline of everything that we covered. All right. Good. So now we're going to move on to that sort of, with that kind of ends the discussion of what macronutrients are. All right. And we're going to move into micronutrients. What's the big deal with micronutrients? So, this unit, I'm only going to be able to talk, I'm only going to be able to have 4 more lectures, I guess, maybe 5, over this. Oh, on Friday, we have a guy named Jeff Wolfowitz from Heiwa Tofu coming to talk to class. So I won't be lecturing them. And I'm going to, well, I'll keep it a surprise. You'll learn in a couple minutes. But before we dive into this broader, more rambling discussion of micronutrients, I have to define a few things. And principally, I have to define what a cofactor is. So let's look at what a typical enzyme looks like. You can have the big blue thing is an apoenzyme. Apo meaning it's inactive. It's inactive. It's not able to do whatever it's supposed to do. Maybe it can't bind the substrate, or maybe if it can bind the substrate, it's not able to do the enzymatic activity that it needs to do to the substrate. Apoenzyme means inactive enzyme. For it to be active, it may have to bind to either a cofactor or a coenzyme of some sort. And if it binds to one of those two or both, if depending on the enzyme of those, then the enzyme becomes what's called the holoenzyme. Holo, H-O-L-O, is the active enzyme. Once that happens, the holoenzyme is able to bind the substrate, and it does whatever it's going to do from that point forward. It becomes, once it binds the substrate, it's the enzyme-substrate complex, and then whatever catalysis happens, that moves forward. But cofactor and coenzyme are two things that could bind to an apoenzyme to make them active. All right? See a couple pencils. Scribble, scribble. I haven't uploaded this yet because I've just sort of finished it, but I'll upload it right after lecture. So here's the same thing, but in a schematic flow for you. Kind of the unit, the semester thus far, I guess. At the top is nutrients. So what is a nutrient? It's something that provides nourishment necessary for the growth and maintenance of life. You have to eat nutrients or you're not going to survive. All right? And nutrients themselves can be broken into two categories. We have spent a large portion of the semester talking about macronutrients. These are things that you need to eat in large quantities, and they are the lipids, the sugars, and the proteins, the fats, the sugars, and the proteins that make up the vast majority of the food that we eat. Then there are micronutrients, and micronutrients are some sort of element or small organic substance of some sort that you need to eat in trace amounts, small amounts. You don't need a lot. In fact, large amounts of micronutrients can be toxic in some cases. But they are essential for the normal growth and development of an organism. So micronutrient is essentially synonymous with cofactor. All right. They are kind of the same thing. Micronutrient, they're just different ways of looking at the same thing. A micronutrient is the way you think about it when contrasting it with a macronutrient. A cofactor is the way you think about the same thing, but in terms of what it's actually functionally doing with the enzymes that it's interacting with. So what a cofactor is doing is it's binding to some sort of enzyme and altering the activity of that enzyme. They break into two categories, vitamins and minerals. A vitamin is some kind of organic molecule, meaning it has carbon, small organic molecule. It's going to have carbon backbone of some sort. And then it may have other elements in it. It may have nitrogen, likely has nitrogen, oxygen, other elements, but it's based on carbon. And then minerals are any of the other elements in the periodic table besides carbon, iron, calcium, et cetera, et cetera. So coenzymes tend to be vitamins, all right? The bulk of vitamins are going to be coenzymes. These are factors that are not going to be tightly bound. They tend to be transiently bound. They can bind and alter the functionality and then unbind. All right? And we're going to see a couple examples. In fact, we already did see a couple examples when we were talking about metabolism, although I didn't stress the role of NAD plus and FAD when I was talking about those processes. But those are coenzymes as well. And then there are prosthetic groups. So a prosthetic group is some kind of non-protein group that is pretty much a permanent part of the enzyme. And they tend to be the minerals. All right. What happens is that, we've talked about how the inside of a protein can be hydrophobic and the outside is hydrophilic. But there can be other things that can guide the folding of a protein. And often that is you can have some sort of metal like zinc or calcium or whatever. There's a lot of different minerals that forms the center, and then a bunch of hydrophilic or charged amino acids are going to coordinate around that, and the protein condenses around that metal core. So these are prosthetic groups. If you were to take that mineral, that metal ion out of the protein, the whole protein would be denatured and collapse. It wouldn't be able to fold properly. All vitamins are not coenzymes, and all minerals are not prosthetic groups. They can be interchanged, right? So a coenzyme just means that it's transiently bound, and a prosthetic group is not transiently bound. You can have vitamins that are permanently bound and minerals that are transiently bound as well, but those are less likely. I have smaller arrows there to indicate that. All right, you follow that overview there? All right, so let's see a couple examples of each of these. Here is some vitamins in the top. So we know that they're organic compounds, and let's look at some examples of vitamins as coenzymes. So these are ones that are either transiently or can be permanently bound. On the left-hand side, we see FAD is permanently bound to whatever enzyme that might require it, whereas on the right-hand side, NAD plus can bind to an enzyme and then can unbind to the enzyme, or it can change its oxidation state. It can go from NAD plus to NADH. It basically is an electron electron carrier is what it is. And it can bind and unbind from the enzyme and participate. And it can be an intermediary between multiple enzymes carrying electrons around. On the bottom, here is a picture of carbonic anhydrase. It's a polypeptide that we talked about when we were talking about the alkaline tide and the production of HCl. And it requires zinc. And you see there that on the left-hand side, it's active with the zinc in place, holding that alpha helix in place next to the beta sheet. And on the right-hand side, the zinc is not there. And I don't know. I mean, this is just a cartoon. I'm not sure whether the zinc is important for global fold, like if that alpha helix would spin out of position, or whether the zinc is actively part of the active site. I'm not really sure about the biology of carbonic anhydrase, but there's just a cartoon example of it. All right, so where do we get all these? They come from the diet. You can either pay lots of money to eat a pill that's going to have all of these different vitamins A, B, C, B complex, C, D, E, K, etc. And all of the minerals, on the other hand, they're iodine, magnesium, zinc, selenium, on down the long list across the periodic table. Or you can just eat. We're fortunate to live in a country like the United States and have access to a dining hall like Foss, Dana, or Bob's that has such a panoply of diverse foods that we can get our nutrients from. All right, so you get them in the food that you eat. Do I want to go through this? This looks kind of dry. I probably should have cut this slide out. It's important, but I'm going to skip this slide. And I'm going to start talking about Popeye. So now, for the rest of the semester, I'm pretty much going to be talking about Popeye in one way or another. I'm going to use Popeye as a way to talk about micronutrients, because as I was putting this lecture together, it's like, oh yeah, Popeye eats spinach. I was looking up something about spinach, like, oh my God, Popeye is far out. So there's a lot that we can learn from the story of Popeye about micronutrients. And please be patient with that. For those of you who don't like Popeye, I can. There is Bugs Bunny in here as well. Maybe that'll be more to your liking. All right. Oh, yeah, right. Here we go. So, does anyone not familiar with Popeye? Okay, good. Let's see. Come on. Hello. Come back. How do I get you to go? Oh, of course. Well, this is why I should have used GIFs. Huh. How do I get it to happen here? Stupid.
Can you just like open it in Safari or Google?
I guess. Yeah, I guess I can. It's whatever. It's Popeye beating up Bluto and eating spinach. It's not crucial to it. Okay, well that's something I'll have to change in the future. I don't know why it's not happening. It's okay. Doesn't matter. So essentially, Popeye's getting his butt kicked by Bluto. It's a real sad scene. He rips open a can of spinach, chugs it down, boom, giant muscles, and there to save the day. So let's look at spinach a little bit first. This was the marketing of spinach around the turn of the century, around the turn of the 19th to 20th centuries. All right, and Spinaceae oleracea is the province and concern of women. Right, this is probably not stuff that men were thinking about. Spinach was a very feminine thing associated with being in the garden, spending time, with baskets. There's a couple cute Dutch kids over there. The cornucopia celebrating America's farmers and gardeners, 225 years of service by Landreths, etc. So that's 1909 to 1911. From 1784 or whatever. All right. And we got from there to this, the manliness of mushy greens. There's nothing like spinach to give a man strength. And here's a Del Monte ad. If you could see most husbands' lunches, you would blah, blah, blah. So now a nice pile of mushy green stuff with some eggs in there, making up for the gross power lunch that most men in ties were eating back in the day. The shift in spinach is rather remarkable. All right, how and why did all of this happen? Well, certainly Popeye was a part of it. All right. To talk about it, let's talk about iron first. Let's begin with iron. Maybe it was the minerals, Popeye says. Here's an early clip of Popeye showing the Rock of Gibraltar in his muscles after chowing some iron. Turns out, or chowing some spinach. Turns out iron is the earliest food supplement. It was used to treat anemia since the 17th century. And in 1832, this guy named P. Bloud, of which I could find very little information. I found some examples of his iron supplements online, but I couldn't find a picture of him or even very much information about when he was born or died. But he came up with a formulation of iron sulfate that was very popular in the treatment of anemia. Here's a package of Bloud's improved iron pills. So this is not actually made by Bloud. He was from France. His formulation was taken up by several different drug companies. This one was formulated by T. Eaton Drug Company from Canada. They improved it by adding 5 grains of Bloud's iron, to which was added arsenic acid, 1/100th of a grain. I'm not sure adding arsenic to the formulation really improved it, but they do say that it was poison on the bottom of it. I don't really know why someone would take a poisonous pill to make them feel better, but this is at least it was truth in advertising. Give them that. So there was an early recognition that iron was important, all right, because anemia was a really pervasive malady for people for a number of reasons, which we'll cover in a bit here. So I'm going to tell you that there's a pretty interesting story around this, what is a myth, that spinach, being the richest vegetable source of iron, which it's not, and this is why E.C. Seeger, who was the creator of Popeye, had Popeye wolf the stuff for strength. Well, this turns out to be a myth, but we're going to find out why it's a myth. And we'll start by looking at how much iron we actually need. So, you need, when you're looking across this, you'll notice that children tend to need a little bit more iron than adults, first of all. And that shouldn't be surprising. Why would you guess that that's true? Why do children need more iron than adults? Because they're growing faster. Are there any other trends on there that you can notice in that chart? Take a look at that data. I don't want to tell you what's what. So compare male to female through childhood up till old age, and what do you notice? Say it. Let's go ahead. I don't say it. Who? Females need more iron. Specifically, what age females? 14 to 50. Why is that? Come on. They're childbirthing years, yep, but not all women have children. Think some more. What else happens during those years? And menstruation, right? So then what's up with 51 years and over? Men and women align themselves once again at that point. Menopause, all right? So at the advent of menarche at the age of 14, women's iron requirements go up significantly to the point where an average adult woman in her midlife has over twice the requirements of iron as a man. And then by the time menarche rolls around, there is I'm sure, welcome relief from the need to have so much iron in your diet. Also, let's compare the need of iron during pregnancy and lactation. Wow, it's a lot. And so these numbers that you see there, the 27 milligrams and the 10 milligrams, just because you're lactating doesn't mean you need less iron. That is in addition to the iron that a normal woman would need, right? So a woman who is pregnant would need 15 plus 27, all right? So whatever that is, 43 or 42. If that woman is on a second child and she's lactating still during pregnancy for the first child, because that does happen, usually women, they may not dry off until right before the second child is born. You're talking about in the 50 milligrams of iron a day. And I can tell you from my own family's experience, when my wife, who is it? Does anyone know Denise Bruzowitz? You do. But she's in the environmental studies department. You know, she's a pretty energetic woman, maybe not running around as fast as me, but she's an energetic person. She definitely doesn't sit around ever. And when we were in New Zealand, and she was pregnant, she during the second trimester, she could not get off the couch. She was just totally weary. Didn't have any energy whatsoever, and she was taking. I don't want to anticipate the story that I'm going to tell here too much, but we eventually got some iron in her, and from some lamb chops, and then she was up and at 'em after a couple days. She just needed some bioavailable iron in her body. So it's real. All right, let's compare. Spinach, 100 grams of spinach has about 2.7 milligrams of iron. You're looking at about 100 grams of spinach there. You know, a small side salad of spinach is going to give you, if you are, say you need 10 milligrams per day, that's about a quarter of your iron. And when you compare it to these other vegetables, it is pretty high in iron compared to many vegetables, like a potato or a raisin, comparable to a raisin, but can't stand up to parsley or soybeans. All right, so already I'm starting to smell something strange here. Why didn't Popeye eat soybeans or, pop a can of parsley or something like that? Why was it spinach? The iron thing is already, I'm starting to question it. And then there are meat sources of iron as well. So we have beef that is roughly half the iron density of spinach. Lamb chops are about equivalent, about 2.1 milligrams of iron per 100 grams. And then pork is about a quarter of the iron density of spinach. All right, and this, if you look at the color of the meat, white meat versus red meat, you can see the correlation between iron density. Well, so what? All right, was he gonna chew on a lamb chop before beating Bluto? I don't know. Well, if we're going to understand why the source of iron matters, we got to understand how the body assimilates and uses the iron. So let's compare spinach and lamb chops. They have essentially the same iron density. All right, you can get the same number of iron atoms out of spinach and lamb chops. All right. First, let's think about how this happens. So your daily intake is, unless you are pregnant or something like that, and should be eating more iron, your daily intake is around 10 to 20 milligrams of total iron in a day. Of that, you absorb only a couple milligrams of that, typically, most people. There is a protein called transferrin that binds iron, and that's going to bind up about 3 milligrams. And then we lose about 1 to 2 milligrams of iron per day. And this happens, this is not in our feces. If you have blood in your feces, you should go see a doctor. This happens by desquamation, that is the sloughing of cells, dead epithelial cells. That can happen off the skin. That can happen on the intestinal mucosa, the lining of your stomach. Those are our epithelial cells. You do slough them off into your gut. Obviously, women lose significantly more than that during menstruation. And on average, people do cut themselves and lose a little blood every once in a while. And that's another way that you can lose iron. Iron is so important. It is a trace element. If you remember back to my tricky question on the midterm, but it is a trace element. But it is so important that the body has developed a number of mechanisms to really effectively hold on to the iron. It doesn't want to get rid of the iron. We have access to so much iron in our diet now because of our access to all this meat, but it didn't used to be that way. Your access to meat was episodic, right? You took down some kind of giant furry thing and ate it, and then you may not have had meat for another 6 months. So over the millions of years of evolution, they have developed no physiological means of iron excretion. If there's iron in your body, we don't really have a good way of getting rid of it. Actually, if you need to excrete iron, there really isn't a need to excrete iron. There's no way to get rid of it. Your kidneys won't get rid of it. Your gut doesn't get rid of it. You have to lose it either by blood loss, menstruation, or sloughing skin. So what happens to this iron that we absorb via the transferrin? About 75% of it gets bound up in hemoglobin. And we're going to talk about what hemoglobin does in a little bit. This happens via what's called erythropoiesis. The making of new red blood cells in the bone marrow of our long bones, and then as you're growing as a child, and then just in the flat bones of your body as an adult. About 10 to 20% of it gets tied up in ferritin. It's a storage protein in the liver and heart. This is where our iron reserves are. You store iron in the liver and the heart. And this is why liver and heart is actually quite nutritious meat, and it's part of a dog's diet. They put that in dog food often. And about 5 to 15% of it is tied up in myoglobin. So myoglobin is like hemoglobin, but it's not in the red blood cells. It lives in your muscle cells, specifically what are called the dark meat, right? And we'll talk about what that means. It's what makes dark meat and white meat different from another. But it's because of the presence of myoglobin. It's a iron binding protein that acts as oxygen storage in the muscles, and we'll see how that works in a bit. All right, so you can be iron deficient. And there are different levels of iron deficiency. All right, so a normal person has a non-deficient person has enough iron for both transport and function, right? So the hemoglobin is functioning, the transferrin is functioning, etc. But then they also have ferritin, the storage molecule that is in the liver and the heart that I talked about. That is full of iron as well. When you get iron depleted, you may not know that you're iron depleted. You have all of the iron necessary for functional purposes, but you don't have any iron bound in the ferritin that's in storage. All right. So your body's able to do all of the things that it needs to do, and you might not realize that you have a low iron level. Then you can become iron deficient, and this inhibits your ability to go through erythropoiesis, the formation of new red blood cells. All right, and this is where you'll begin to notice it. You'll get out of breath a little bit easier when you're running upstairs or something. You'll be a little bit more weary. And then you can move to real iron deficiency or anemia. And this process happens a lot more rapidly for females. We'll see that in just a minute here. So the average adult male who starts out as normal, if you were to start with the full complement of ferritin storing iron in your body, you would have enough iron to keep going for 3 years. An average adult female who is not pregnant but who is menstruating normally would only be able to make it about 6 months. And this is because so much of the iron in the woman's body is depleted each month during the process of menstruation. So this is the biggest loss of iron, biggest chronic loss of iron. Oral contraceptives can actually decrease that and lengthen that amount of time that a female could go without iron. And intrauterine devices, IUDs, have the opposite effect. Pregnancy obviously is going to significantly impact that, as breastfeeding as well. As a baby, a child growing into adolescence, your need for iron may be higher. If you're a blood donor, you need to be aware of replacing the iron that was taken out of your body after the removal of the blood. One of the things you can do to increase the amount of iron in your diet is cook with cast iron. Does anybody like to cook with cast iron? Anyone? Yeah, it's great. We have a new induction stove. Cast iron is lovely on an induction stove. So yeah, cook your tomato sauce. Cook acidic foods in a cast iron pot. It can. The statistic is you can increase your iron content of your food by 80% just cooking in a cast iron pot. All right. So, what is it really doing in the body? Well, the biggest thing, we're going to talk about hemoglobin, ferritin, and myoglobin, each in turn. First is the hemoglobin. Hemoglobin is how we get oxygen from our lungs to our tissues. The oxygen is brought to the pulmonary surface as we inhale during ventilation. The red blood cells go to the lungs and pick up that oxygen and bring them to the peripheral tissues, where they're used by those tissues for internal respiration. That's the metabolism that I talked about last week, or a couple weeks ago, whatever that was, last week, I guess. During when the mitochondria is going through the TCA cycle and electron transport chain and producing all that ATP. All right. So let's see what's actually happening by looking closer at a red blood cell. We zoom in. A red blood cell is packed with millions, in fact trillions of hemoglobin molecules. As we zoom in, we see in the middle of this hemoglobin there are 4 subunits, and each of those subunits has what's called a porphyrin ring in it. It's this complicated organic structure that you see there. You certainly don't need to memorize that structure, but it kind of is like a trampoline. It's like a molecular trampoline with an iron sitting right in the middle of the trampoline. All right? It is that iron that binds the oxygen. So you can see the trampoline on the side here, and it's tethered to the hemoglobin molecule by this histidine residue that attaches to the iron. And then the iron is able to attach to the oxygen. We all know that iron binds oxygen. Here's an example. Here's this sculpture by this guy, Julian Voss-André, who it's called Heart of Steel. It's a model of the hemoglobin molecule with a big iron in the middle. And with time, that brushed iron rusted. It's probably a nice rusty mess at this point. But that was over the span of 3 years. That's what happened to it. All right. So each red blood cell has about 280 million hemoglobin molecules in it. There are about 30 trillion of the red blood cells in the body. Do the math. That's 8.5 times 10 to the 21st atoms of iron per person just tied up in hemoglobin. Hemoglobin itself is an excellent source of iron in the diet, then, principally through meat. So, for example, does anyone... red blood cell. Sorry. Has anybody been to Ireland or Britain or anything like that? Did any of you ever have blood pudding before? No. A black pudding. Maybe you did if you had breakfast sausage, because it's what giant scabs. Nope, they're essentially giant scabs. They are so yeah, sure they are. They're tasty. I've had one. Yeah, in Ireland they didn't have access to a huge amount of meat, but they had a high need for iron, like everybody does. And so what they would do is they'd go out to the cow or the goat or the pig, and they would bleed it a little bit without having to kill the animal. They would bleed the animal, then take that blood and go through a long process. Here's the recipe. I apologize to all the vegetarians in the room for turning your stomach with all this. But you take 4 cups of fresh pig blood, add a bunch of salt, add some oatmeal in there to thicken it up, a little bit of onion, etc., some allspice just to sound nice, and they would make it into this sausage that was an adequate source of iron for them. It met their iron requirements without having to actually kill the animals that they were making it from. All right. So, all right, yeah, I just started talking about a lot of meat there. We were trying to understand why spinach is the food consumed by Popeye. Well, turns out that the body is able to absorb iron that is bound up in hemoglobin much more readily than non-hemoglobin. All right. So if you take in 20 milligrams of iron, the iron in that 20 milligrams that is bound up by hemoglobin in the meat that you eat is maybe 2 to 3 milligrams. And the non-heme iron is 18 to 19 milligrams of that 20 milligrams. So 10 to 15% of the iron you consume comes from meat, and the intake, and 85 to 90% comes from vegetables. This is what you eat. What you eat is a different thing to what you absorb, right? So let's look at the difference there. So now if we look at how much you absorb of that 20 milligrams, you're only absorbing about 2 milligrams, okay? We had said that earlier. And of that 2 milligrams, about a milligram's worth, about 0.7 milligrams worth, comes from iron that was bound to hemoglobin from meat sources, whereas the non-hemoglobin is only 1.3. Still, 60% of your iron is coming from vegetable sources in a typical diet, and only 40% from meat sources, but the efficiency is 5 times higher, 35% efficiency of intake of iron from iron that is bound to hemoglobin, and only 7% efficiency. Most of the iron that you're eating in vegetable sources is passing out of the body in your feces. And why is this? This has to do with the solubility of iron in water. Iron that is soluble, iron that is bound to hemoglobin is much more soluble than iron 2 plus versus iron 3 plus. These are different oxidation states of iron. We'll talk about that in a bit. And if it's soluble in iron, if it's soluble in water, that means your body can absorb it and pull it across the membrane into the cells. Okay, so that's where we'll leave off. I'll pick it up here next time.